welcome to Inside the Onion, the podcast where we peel back the layers surrounding a given topic within science, society and culture, examining it from many different angles, presented by myself, Angela Pun, and my co-host, Joe Clerk. Joe, what juicy onion are we slicing into for today's episode?
So Ange, today we will be taking a deep dive into cephalopods, the family of species containing squid, octopus and other tentacly things. We'll be particularly zooming in on the deep sea squid, as today we have an expert on them and their dietary preferences. A warm welcome to the show to Laura McBride. How are you today?
I'm very well, thank you. Thanks for inviting me. It's so funny you call me a squid expert. I feel like I'm an amateur squid expert, or
you got a paper out there. You're an expert.
Yeah,
right?
or an expert in training. But I'm really excited to be here and get into talking about squids and other cephalopods. My name's Laura McBride and my pronouns are she, her. And I have an undergraduate or a bachelor's with animal biology, where I met Angeli. And then I did my master's in Groningen University and did ecology and evolution.
Fantastic. Well, first, for those of us who aren't experts in cephalopods, could you please tell us why we should be interested in them and also how you came to be involved in this area?
So I feel like the actual professional academia answer is that they're just such an anomaly in themselves, and the fact that they're invertebrates and they're so incredibly intelligent and they're everywhere in all the world's oceans, and we still don't really know that much about the deep sea squids. My personal opinion of why we should be interested in them is because they're so cool and they're so cute.
That's such a thing in academia. It's like you have that answer and then you have it's just so interesting.
Yeah, literally, because my supervisor for I did a presentation on them in university and my master's about the cephalopod intelligence, and she was like, oh, so why should we care about them? And I was just like, they're so amazing. She's like, that's not the answer I was looking for. And how I became interested in them, so in my undergrad I did an internship at St. Abbs Marine Station through Dr. Karen Deal, and it was looking at how anthropogenic noise affects squid embryogenesis on Loligo vulgaris, so common squid. And so we were playing...
What's anthropogenic noise?
Yeah, sorry, I just totally rattled that off. I've said it so many times.
Right.
So anthropogenic noise, so anthropogenic is anything that's human caused. So anthropogenic noise would be things like ships, building wind farms.
Okay, but we're talking about literal noise, like sound.
Sounds in the oceans, sounds in the seas.
Caused by humans.
Embryogenesis is how animals develop in the embryo or in their eggs. So what we did is we had 2 tanks. One was completely just silent, and we had squid eggs from their first conception, and then the other tank had ship noises playing in them, and then we would take the eggs and look at them under the microscope and see how they were developing in comparison to each other. Was one developing slower than the other? Were there more deformities? And we would do this over a period of weeks to see the changes of them. And then
what did you find? Was there a difference?
Well, I was only part of the study for a little bit, and then the study got cancelled, so I actually don't know. So if someone wants to pick that up or give us some funding for that would be great.
I think it's interesting because it's something that isn't thought of so much in the whole humans affecting the climate is noise. We'll think people will commonly go, okay, well, changing temperatures, that affects animals' habitats, and that affects this and that. But there's actually so many ways in which our activities can affect the natural world that we need to be considering in this moment in time.
Absolutely. I mean, up until recently, people thought that the sea was silent. And then we found out that it's very noisy. It's obviously just at frequencies that we can't hear. There's fish that communicate with each other all the time. Obviously, you've got echolocation and cetaceans and stuff. So it's kind of trying to get over that misconception as well. But St. Abbs, and it was Edinburgh Napier University, and I think now Edinburgh Uni have been doing loads of research on it. So that's how I kind of got initially into it, and then I kind of did a little detour, wanting to kind of focus on land-based animals, I guess, or terrestrial animals. And so how I really got into squids and doing it in my master's research, I was in Sweden working on another project on pied flycatchers, and I went to bed and had this dream that I was going to be a squid researcher. And then I woke up and I was like, well, that's what I'm going to do.
Dreams happen for a reason.
Right.
They're not, dreams aren't random.
I know. I feel like it was like a subliminal message coming to me.
So yeah, what are you up to nowadays then?
So currently I'm working as an associate scientist doing Sanger sequencing and next generation sequencing on viruses and bacteria, and but not really ecology based. But later this year I'll be starting a PhD in Aberdeen.
Right, congrats on getting that.
Thank you very much. It's not unfortunately cephalopod related, it is fish related, so it is another detour, but it's fine.
So I guess before we dive into the various layers of this topic of cephalopods, just a quick disclaimer. We'll briefly mention here at Inside the Onion, while we're going to be broad and cover each subject, or each onion as we call it, from a variety of angles, of course we can't cover every angle of every subject by any stretch, and we will probably skip over some things, but we hope, however, that you, your listeners, can get excited about them as we are from learning from them from this podcast. So I guess perhaps we should first be clear on what exactly we mean when we say cephalopod. What exactly is a cephalopod? And here I'll probably again defer to the two marine biologists in the room, as I feel like they're best placed to answer this question.
Yeah, so a cephalopod is a class of mollusk and contains octopus, squids, cuttlefish and nautilus. And I guess kind of their defining features is that they have bilateral symmetry. So that means if you cut it right down the middle, the two halves will look exactly the same. And also characterised by having many arms or tentacles, a head and then a mantle.
How long do these cephalopods live for? Is it the same across the class, or does it vary? Or?
Well, interestingly enough, so they're known to usually live about 1 to 2 years, and they're semiparous, which means that they have one reproductive cycle, and usually once they reproduce, they die off.
I remember seeing what was it, the documentary, like My Octopus Teacher.
Yeah,
and it's so just nice. Like it's incredible, but the dying stages and it's so like it's such an intense way to live, you know.
Yes.
It's like,
live fast,
you
die
discard
young.
your babies, else you wither away.
Yeah,
like, it's crazy.
Literal live fast, like die young, and then they just like waste away, like their body just like breaks down, like because all of their energy's been spent into their babies, and then they just stop eating, and then they just kind of like decay away. However, I think it was researchers off of Monterey Bay found a species of Granellodon boreopacifica, and it was a female who had been brooding her eggs for 53 months.
Oh wow.
So Mother's Day presents for her, like what the hell?
Wow.
Which is impressive considering that
Did you say it was typically a year?
To 2 years, and this one was seen not just living but like just specifically
like
guarding her eggs 53
years.
months.
Wow.
That's 4.5. So there must have been some freak genetic mutation in her or something.
Yeah, her eggs were just not.
No, I don't know if it's.
Did they ever find out why?
No, that's the thing. It's just it was such a shocking discovery, because again, that's what we know, and again, it just kind of like blows everything out of the water. She was a deep sea species, but she didn't eat, she didn't move, she was in the same spot. They went back again to check, and they found her in the exact same spot. Like
that's so crazy. Interesting.
Crazy.
So it could be like that specific species could have a longer lifespan.
Wow, that's so interesting. So like I know that like cephalopods are also known for being highly intelligent. We have an example of Paul the octopus, who was able to predict the World Cup outcome in 2020.
2010, wasn't it?
Yeah, 2010, sorry, yeah.
Yeah, they also discovered that, I hate using this term human-like, but like one paper found that Dicidicus gigas, so jumbo flying squid I think it is, have developed human-like syntax.
Oh wow!
Through their photophores or chromatophores to communicate within their group, like when they're in hunting groups, which is kind of interesting again because normally squids are
normally solitary.
Solitary, or they're like, if you see them together, they're like cannibalizing each other.
Wow, that's so
but they're hunting in groups, and then they're able to literally like communicate like grammar and syntax to each other through like flashing of lights.
But I mean, you see all these videos of like squids like opening or octopuses opening jars and squirt. I remember there were stories of like octopuses squirting jets of water at researchers. They're just like mischievous and like got all these weird qualities,
like little personalities.
That's so interesting that they're able to like also though communicate, because that's like mostly like a mammal thing. Like I know that dolphin and whales do that. They communicate with conspecifics to like for maybe like hunting or just like kind of keeping within a pod. So it's interesting to hear that.
Yeah, I've been doing a bit of digging into the nervous systems of them because that's always going to be my thing, and like it's fascinating. You know, whereas like we as mammals have these like centralized nervous systems based around a brain, and then we've obviously got a peripheral nervous system that allows us to control our body and feel things through sensation, but it's far more distributed in the cephalopods.
Oh, for sure. Well, I mean, like each of the chromatophores are like controlled by a single neuron to like control the color, the shape of the cell to then change the color of the whole. Within a second. As soon as you think about it.
Is that just octopuses that change colour, or
squid, cuttlefish?
Right, okay.
I don't know about nautilus, but cuttlefish especially, there's lots of research on them, on their like mating patterns and stuff. They'll also I think that there was one observed that one was trying to flirt with a lady cuttlefish, and then saw like another male competitor, and then changed one side of the pattern to show like a lady pattern to the competitor male, and then on the other half had his like mating pattern towards the female.
Incredible.
And the fact that they're so cute, just thinking about like this sneaky cuttlefish just trying to deceive cuttlefish. Oh, that's so adorable. But yeah, also deep sea squid have bioluminescence possibly. Is that a thing that they use for like hunting? I know that like a lot of animals in the deep sea use bioluminescence as like some sort of like lure for prey.
So like what's the classic? The anglerfish is the one that does that with the little in terms of luring prey. I don't know about they catch bioluminescent things.
So they have the light bulb in front of
the little stick in front of them. I didn't know the squids lured stuff in. Okay, that's cool.
Yeah, so like I guess like squid and octopus are able to like use it for like I guess like attracting mates, for camouflage or like warning signals, and like even to communicate, as you said before.
Speaking of eating, you've got written down here that they feed using a beak.
Yeah, they masticate their prey till it's like indistinguishable. They'll just like, if it's like too big for them to swallow, they'll just like destroy it.
They're just chomping away.
But if you've seen like a picture of like a Architeuthis, which is like a genus of squid, their beaks looks like you would see on like a bird of prey, like an eagle or like a falcon. It's got that kind of
the curved beak.
Yeah,
to like hook onto things. And it's like, Oh, wow.
Because you think we just sort of think of these as like squishy things, but I guess is that the only bit that's not squishy, the beak, or
so they have so cuttlefish bones, it's not really bone, it's cartilage. That kind of keeps their structure together, I guess, like kind of how we have a spine that keeps everything together in shape. And then squid have gladi, which is kind of like it kind of feels like a papery bag, like a plastic bag, but it's got like a bit of like structure to it as well that keeps them in place.
So these animals are amazing. we can like obviously discuss their like amazingness and biology further, but or we can even do a whole podcast on that. However, since we have Laura, our guest, in who is a squid expert, maybe we can talk about her research. But first, let's just debunk a question. So I've always wanted to know: are octopus and squid, are they actually aliens or?
Well, my friend Sam Crehan told me said a really interesting or profound thing I'd like to say, is that we like to consider them as aliens, right? Because they seem so otherworldly. They look like, like stereotypical like pop culture aliens, and they have all this mad intelligence. And
despite I read their common closest ancestor, or at least octopus's common closest ancestor of this was the sea cucumber.
Oh really? I didn't know that.
So I mean, like, it goes that
we're so far removed.
Yeah. And yet they show traits that are, like the mischievous behaviour, like the complex developed nervous system, things that we think of as like, oh, that's what a mammal does.
Yeah.
Right? Yeah.
Things that we think are exclusively mammalian.
Yeah.
But then you think of like bird intelligence as well, and obviously their brains are developed differently. I mean, obviously you can have like a whole other side chat about like, that intelligence isn't just a mammalian trait, and different branches of intelligence. And then who's to say that our intelligence is considered intelligent by an octopus or a squid, where an octopus could be like, oh my god, I can fit into this tiny hole and you can't, you must be so stupid. But if you think about how life on Earth has evolved, we all started from the deep sea and then gradually moved our way onto terrestrial landscape. So if you kind of think about it, we're the aliens.
Yeah, no, for sure. They stayed in the sea, so...
I'm kind of alright with being an alien. That's kind of cool.
I mean, sometimes I feel like I'm an alien. Like, I'm just like, why do I not just get on with these people? Am I weird? Yes, I'm definitely weird. Who knows?
So we need to know, are they tentacles or are they arms?
Right, so octopuses don't have any tentacles. They have arms. And squid and cuttlefish have 2 tentacles, and the rest are arms.
So what differentiates a tentacle and an arm?
It's the suckers on them. So tentacles, if you think of like tentacles as like arms and hands, the hands are the only ones that have suckers on them, whereas well, that was not a good analogy.
Yeah, I'm more confused now.
Right, so if you think of a squid, and you have the arms, and then you have the tentacles, and all the way down the tentacles is just smooth, I guess.
Okay.
And then at the bottom, like in their little like hands, I guess.
They're like paddles.
Paddles. That's where the suckers are, whereas on their arms, there's suckers all the way down.
Okay. And why do they have a mixture of tentacles and arms? Do you know? Or...
I guess probably just for catching prey. I mean, like one's...
So what, tentacles with the suckers on the end are better for just sort of like grabbing
and then bringing to your mouth.
Right. I know that is like one like adaptation for like deep sea squid, is like they have like longer tentacles because it's easier for them to kind of gather prey.
What is it? The large fin squid. They look horrifying, but they look really cool, and they've only kind of recently been picked up on like oil rig cameras, and they just have these like big, long, dangly arms, and they don't do anything with them. They kind of just they theorize that they use them just to like waft about and wait for like things to swim into them, and then they bring it up to their mouths.
Right.
Oh wow,
there might be some sort of sensory organs going on in there to help them navigate. Well, I guess maybe they use them to detect some sort of waves in the
good for you,
water.
Almost like a Venus flytrap, I guess, or like something like just there waiting and then for something to touch it and then trap.
Or if you think like a jellyfish as well, where like they have like the stingers and then that paralyzes their prey when it swims into them, and then they eat from that. But although nautiloids don't have arms or tentacles, they have
jellyfish. Scare the hell out of me.
Siri.
So they have like the little the face beardy thing.
Yeah. So they have loads of them and no suckers.
Yeah. Wow. Interesting.
The sea's a crazy place.
It is a crazy place.
It's a crazy place.
It is, and it's easy to see why they're considered aliens, because like if you look at quite a lot of deep sea creatures and just sea creatures in general,
it's like
just
my favourite episode of Blue Planet is always the like freaky things at the bottom of the ocean that are glowing. Like it's just so cool.
Sure.
It's so
and
cool.
just how they've adapted as well.
Yeah, it's crazy how things adapt to like such extreme conditions.
Mm-hmm.
You know.
Shall we talk a bit about your research? Me and Ange have both been checking out your paper and now going to do a rehash of your master's thesis defence. Have you ready?
Oh my god.
Nah, nothing of the sort. So I've got the paper in front of me here. Trophic ecology of the deep sea squid Mortothopsis ingens. Is that right? Yeah.
Well done.
Otherwise known as the warty hooked squid from Chatham Rise, Aotearoa, New Zealand.
I think it's Aotearoa. But again, I could be saying that wrong. That's the Maori term for New Zealand.
Got it. Well, I managed to mess up pretty much every single stage
That's
of that title,
fine,
so
don't
you
worry.
can save me with. If you could just give us a bit like the abstract, if you like, what it was that you were doing out there in New Zealand and what you found.
So there's an area off the east coast of New Zealand called the Chatham Rise, and it's ecological and economical importance, as well as having really strong cultural ties to Maori culture as well. And we're just kind of trying to get like an understanding of the trophic ecology of that area. And
when you say trophic ecology, just for those that aren't in that field, like
so it's about food webs essentially, and how food webs are built and how energy is transferred through the columns and levels of animals in the area. Okay,
cool.
Yeah, so Mortothopsis ingen, or the warty hook squid, is one of the most common or prevalent species of squid in the Chatham Rise. So we wanted to understand more about its trophic or like feeding ecology. So previous studies have only really focused on visual identification of prey species, and so we wanted to have a more robust understanding by introducing DNA barcoding. So using Sanger sequencing and using CO1 barcoding. So CO1 is a gene, which it's a mitochondrial gene. So if anyone remembers from high school, the mitochondria is the powerhouse of the cell. It's involved with energy transfer. It's very important, and it's expressed in all animal species, and it's really easy to amplify. So that's usually used as a marker of identifying species.
Mm-hmm. It's mitochondrial DNA that was being sequenced.
Yeah, basically it.
Interesting.
So it's used in like cellular respiration and stuff.
Mm-hmm.
And you were sequencing, so their stomach contents, right? You take the stomach contents out and sequence what you could, what had been completely obliterated by the heavy chewing that we discussed earlier.
Yeah, so that's why visual identification can be quite difficult, because squid like to massacre or like absolutely obliterate their prey items to fit into their beaks. So it's not really always easy to rely on. Some things you can use through visual identification. So things like sucker rings can be unique to each species of cephalopods or squids, and otoliths as well, which are tiny little inner ear bones in fish. And otolith shape is unique to each species as well. So you can determine what species of fish.
How small are these? Like you talk about inner ear bones of fish.
I mean, that must be so fiddly to
They're
play with.
so
I kind of love doing fiddly stuff under microscopes though. So did you had little mini microscopes to check, and then just manipulating them with forceps and things like that, or?
So I did my research at the research group ALSACE. So that's Auckland University of Technology's lab for cephalopod ecology and systematics. So shout out to them.
Nice.
And we got help from Darren Stevens, who is a part of NIWA, which is the National Institute for Water and Atmosphere based in Wellington. And he helped us with otolith identification because he's like a wizard on that.
Amazing.
He's like the expert on that. So
there's always someone who's got their like specific niches. For
I love how academia is just filled with people who just have the weirdest, super specialist expertise.
Like,
just like, oh, I know this guy, the fish inner ear bone guy. And then we also had this PhD student. She was studying at Otago University called Jerusha. And she is an expert on parasites as well. So we found like quite a lot of like parasitic worms encased in the squid stomachs, which are absolutely disgusting.
I bet.
Like even thinking about it makes me shiver.
Oh, no.
But she was really helpful on that. So yeah, they were really fiddly. Some of them are like a little bit bigger than others. So the New Zealand dory fish is like a little bit bigger and it's a bit more thicker, I want to say, but some of them just look so fragile that they could just like shatter easily.
And so what did all this inner ear poking and DNA sequencing reveal in terms of how this warty-hooked squid feed in the Chatham Rise?
So we learned some really interesting novel results from this. So we discovered 5 new prey species for this animal. Not species that have been discovered in the world, but just discovered as food for the squid.
Right.
Wow.
So that was really interesting. We saw signs of so these squid species, the warty-hooked, does a lot of like diel vertical migration. So that means that during the day they'll be in like depths of over 200 meters, and then they'll like come up to the surface and feed in the surface as well, and then go back down.
Is that like common in the marine environment to have
I feel
animals?
like that's common. Like quite a lot of animals do that. They follow like the plankton up
Right,
towards the surface and then down again.
Because I guess it must take quite a bit of like pressure adaptation to be able to withstand that. But I guess it helps if you're squishy.
Yeah, well, that's the thing. That's why you can live in like these like unforgiving environments if you don't have bones that break.
Yeah, definitely.
I mean, I don't want to try that anytime soon.
No, absolutely not. It's too dark as well. It's too scary.
Yeah.
So that was really cool. What else did we find? We found Bathyteuthis abyssacola, which is a type of squid species as well, a bathyteuthoid, which had been discovered as prey in a squid for the first time ever.
Wow.
And we found that I think it's maybe juvenile warty-hook squid or is it juvenile bathyteuthoids and the warty-hook squid, their ranges overlap. So we could think maybe like that's how they like interact with each other, because bathyteuthids are like deeper, like normally like the adults like live in deeper water columns, but I think we believe that the juveniles live in like higher levels of the water column. And then we also discovered, or may have potentially discovered, a new species of lanternfish. Wow. So that's really exciting.
That's really exciting. That's really exciting. So when will that be confirmed, or would that be confirmed?
That can take years. It's really hard to get these things rolling purely because you need to fully establish a new species. You need to have the DNA, which we have, but you also need to have the physiological aspect of it, and it's kind of hard to like, in like deep sea like lanternfish that all kind of look the same, it's kind of hard to determine. You have to like look for like
so there'll be a bit of time yet before lanternfish McBride is
Margaret after my mum, because she said she wanted to be named after.
Imagine a lanternfish called Margaret.
I know. Margaret's lanternfish would be so
that's got to be like one of the biggest flexes in life. It's like, oh yeah, I've got a species named after me.
Yeah, she can live forever.
Oh, bless.
So it was quite interesting because when we were looking at prior papers, going back to the otolith morphology, there was quite a lot of otoliths that they couldn't really classify because it kind of looked a little bit too different from one lanternfish, but they didn't have a reference for another lanternfish. So this like DNA barcoding that didn't match any of the reference barcodes in the Barcode of Life, which is this massive database that has all of the DNA barcodes of every documented living thing. It didn't match anything on that, and then we see in previous papers that, we also have this otolith that we don't really know who it belongs to, but we kind of know it's a lanternfish, so it's like, this can corroborate that as well.
It's like you're in a detective case and you've got like little bits of circumstantial evidence that are like pointing all in the same direction. You've all got a hunch, but you need the final bit to be able to convict.
It's like that totally. It's Always Sunny, Pepe Silva, kind of like all the like strings are like connected and stuff.
love that.
For sure.
And then we also saw cannibalism as well, which
interesting.
Isn't that uncommon in squids, but it's still always interesting to find out.
Yeah, I mean, that's what I
just sounds pretty crazy. Like, coming from the outside.
I thought that was pretty interesting when I read about it. I was like, oh, cannibalism, but then I kind of remembered like, like gulls.
Yeah, there's a lot of species that do.
Like, even ladybirds, like they all like...
Not little ladybirds.
I know, they're so humble.
But I guess like broadly speaking, that's like it's quite easy to see at the individual level why, like, you want to carry on your genes, not your competitors. But then at the species level, it's like it's quite weird to think like how that evolutionarily speaking benefits a species. I guess that you said they were one of the most abundant squid species so like probably it doesn't affect in the same way. It wouldn't happen the same way if they were under threat. They wouldn't. Yeah,
I think it's like that kind of self-limiting feedback loop of like these squid have such a high energy lifestyle, and they're just grabbing everything and anything that can like fit into their mouths and that they want to eat and stuff. It's
I mean, I know states like that,
right? And it's so funny when you think of their brains are like donut shaped around their stomachs. Can you imagine if you eat too much and then it actually gives you brain damage?
I know. I was just thinking like maybe they're thinking about donuts because their brain's donut shaped.
Yeah. I think that because they're so abundant that they're going to come across them more often, that's why cannibalism can occur.
Okay. Yeah.
But I guess if there ever a time that their population really diminished, your chances of coming across another species is so little that cannibalism wouldn't really occur, and then I don't know if that would bring...
I mean, it's like also like a competition thing, like kind of, just scavenging, eating what you can, but then also like removing competitors.
Yeah,
I guess
sense.
Like as one of the most abundant species in that region, they're going to have like a really huge effect on the food web around them. They're probably going to impact a whole and I mean the species list that you got and you were saying to us probably doesn't even cover everything because of limitations in techniques. Like, so I guess can you kind of like give an overview of how studying the diet of a really abundant species like this in this case could help to, I don't know, maybe inform conservation work or like how it helps us think about how we approach that region?
Well, you think of where they sit in the food web, they're kind of like in the middle, so they're predated on, but they also prey a lot of things. How they live and how they eat can also determine like how healthy fish stocks could be in the area, which obviously has like an economical effect for that. But then you can also think of like it's incorporating lots of different food webs. It's like a tie between lots of different food webs, from like zooplankton to fish to piscivorous fish, etc. And you think of like all the way of these energy transfers, and like with, I guess if you always want to tie in climate change, it's obviously very relevant, but like understanding how that energy is transferred can help us maybe work forward into how to protect these like this flow if it ever breaks down through climate change.
Yeah, well, like that's one thing as well that I was going to ask. So you said that you'd found 5 new prey that this squid had eaten, and this was like in comparison to the study that was done in 2003. So would this finding be because of like the new up-to-date like method that you're using in terms of like DNA IDing, or could this be that these prey items weren't actually there in the area over like almost 20 years ago, and could maybe like anthropogenic activities or things like climate change be affecting the prey that availability of these squid?
I think one of the main reasons is purely just because we have a more thorough technique of discovering these things. When we find these prey species as well, when you reference their or sequence their barcodes, and then you check the references, and then you find out what's in them, you look at the ecology of that species as well, and you can be like, oh, that's present in that area, that's very common to see. There was one barcode that we got that gave up a very kind of peculiar answer, and we found that it was like a fish species that was present in South African waters. And we were like, that's quite far from where we're sampling from. And we found out that it was actually the barcode on that was wrong.
Oh, okay.
So I wouldn't say maybe climate change has influenced that. That's not to say, from this study, we can't say it's like climate change. But we also, looking at other past papers as well, we were sampling at different parts of the year as well. So there could be like seasonality in fish abundance and stuff and things like that.
No, totally.
But if you want to kind of like maybe measure climate change and its effects, you would have to do it like same technique, same sampling areas over periods of time. But like as someone who would study cephalopods, it's kind of like they'll be fine because they eat anything. They're like really adaptable. Like they'll dominate the seas. Them and jellyfish, it's like it's fine. Which is kind of sad. But obviously, like you can use that as an indication to how maybe species in the lower water column and then the higher water column and even in the like in the air, because these squid, they transfer carbon all throughout the and energy all throughout the water column, and then when they die, because they're filled with like ammonium, and when they die they just like float up to the top, then seagulls eat them afterwards, and then that energy is getting transferred from the sea to land via...
They're feeding everyone.
So actually going back to the ammonium and like their buoyancy, like that is like something that I guess like many like deep sea creatures, like this was something that was like new information to me, that many deep sea creatures use it to like aid their buoyancy. Like, would you want to maybe explain that more to me? Like, I was quite interested to like read that.
Yeah, I found that interesting as well. And it makes sense if you're moving through the water column and obviously at different pressures and stuff. But I think what's really interesting about the fact that the reason that they use ammonium is because it's lighter than sodium in their blood, so that helps that kind of buoyancy. But the fact that also like ammonium is just readily available in their bodies because they have such a high carnivorous diet, it's just a byproduct of breaking that down.
Right.
So the food that they eat contributes to the ammonium in their muscle tissues, which is really interesting. Which is obviously, as I said, really good for daily vertical migrations. Compared to like cuttlefish, some use gas chambers, and then like some octopuses use swim bladders. But it's kind of interesting because, as with anything, there's always a trade-off. So like having high levels of ammonium in your muscle tissue means you have to sacrifice like strong locomotion for like moving around and stuff. But then again, if you're a deep sea animal, you don't really need that strong of a locomotion. Because you're kind of just like conserving energy.
You're not, I guess there's not as much risk of predation down.
You're not as visible. Like if you think of like more shallow water species, they're visible as predators and they're visible as prey.
So they would have less ammonium and have more escape, a bit more locomotive ability.
That's it. Because I guess like I mean like you're gonna have to have all these like special adaptations to like, or adaptations, sorry, to like live in the deep sea and also be able to do these like vertical migrations. You know, like
if we're talking about special adaptations, like the flip side of that with escape, is something that the squid in general, maybe not so much the deep sea squid, although I guess it still has this, is their giant squid axon.
Yeah.
Which I remember from first year neuroscience. It's an incredible story.
Yeah, I remember watching a video about it during my
I'm
So-
not so
undergrad.
knowledgeable on it.
So it's amazing, really.
Well, I mean, it controls their siphon, which kind of...
Yeah, so it controls their jet propulsion that they use for escape. But the axon, so just for anybody that isn't au fait with neurons, the axon is basically the sending part of the neuron. Action potentials, the way the neurons fire, are generated in the cell body or the soma, and then they're transported along the axon to the axon terminal where they release neurotransmitters. And
Which controls movement.
Which controls everything.
Yeah, everything.
It's the way that the vast majority of neurons talk to each other.
Yeah, but I think in this sense, like, it controls the siphon, which helps.
Yes,
It has that quick
in
response
in this case,
very,
and
very
basically
fast response.
conductive, conduction speed of these electrical signals along axons is regulated by many different factors, but one of them is the size of the axon.
Mm-hmm.
So human neuron axon, or even the majority of the axons in the squid, are about 1 micrometer in diameter.
Wow.
Right?
So comparing that to
The giant squid axon?
Yeah.
We're talking about 1,000 times as large.
Wow.
1 millimeter.
What?
Well, the range is from 0.5 millimeter to 1.5 millimeter. So visible to the naked eye.
Yeah.
And that's the main reason why it's so important in a history of neuroscience
I mean,
research.
it's been groundbreaking, you know, like...
Because you could see it with the naked eye. I mean, like nowadays we can do all sorts of incredible things down microscopes.
Yeah.
But and for sure they were still using microscopes on this because it's still only a millimeter wide.
Yeah, but still.
But they were
to...
it was first discovered and described as a axon, as a neuron, in 1909 by this L.W. Williams. I think they first thought it was a blood vessel, but then they realized no, this is like a nervous cell. But it's synonymous with the Nobel Prize winning experiments of Alan Hodgkin and Andrew Huxley. Nice little AH combo.
Shout out to them.
Yeah.
All
So basically, yeah, they attached electrodes to that and saw the action potential firing, and saw by blocking different types of channels, they were able to see what ionic fluxes regulated action potential firing, regulated neuronal firing. So if they applied tetrodotoxin, so this is the toxin that comes from a range of different animal species, most famously probably the pufferfish.
Is that what gets the dolphins high?
Well, yeah, which is crazy. I mean, it blocks sodium channels,
Wow.
which is basically essential to neurons firing.
So it like inhibits them, like kind
yeah,
of like...
exactly. Like an influx of sodium is what leads to the neuron spiking, and then an efflux of potassium is what leads to the hyperpolarization that comes afterwards. And
what's hyperpolarization?
so basically neurons stay at a certain membrane potential depending on the amount of positive and negatively charged ions that lie inside or outside the cell membrane. It typically varies from cell to cell, but in, say, the majority of neurons, you're talking about minus 70 millivolts. And then there are a series of channels on the cell membrane that open at a particular voltage, say a given threshold value, maybe around about -50. So if an incoming signal has I'm sweating now that it's my own stuff.
I know, right? How does it feel?
Yeah, how does it feel? How does it feel exactly? I mean, no, this is 101. But no, if the intracellular concentration of positively charged ions increases relative to the outside to a certain degree, and this could be because certain incoming neurotransmitters have caused their own channels to open and allow an influx of ions. Basically, once the cell gets to that membrane potential threshold, a series of voltage gated channels open. This allows the influx of sodium and in some cases calcium ions, then that causes the neuron to spike to a certain level, and then at that stage, once it goes above 0 to around maybe like +40, +50 millivolts. At that stage, another set of voltage gated ion channels that are fluxing potassium. So a baseline will have a high intracellular concentration of potassium, another positively charged ion. And so when it reaches the peak of the action potential, those channels will open. There'll be an efflux of potassium, and that brings that's a repolarization. The repolarization, hyperpolarization of the cell, and that sort of prevents it for a little bit from firing again. And so this pattern, of action potential firing that is now widely understood to be how neurons operate, was first discovered.
All seen
in the giant squid axon.
Wow.
So they
with the naked, under a microscope, but not because when usually you look at these things, it's like electron microscopes, and it's like.
Yeah, I mean, so I was doing these kind of experiments during my PhD, and we were looking under 40x microscopes with with pipette, patching onto cells, because the axons are so tight, patching onto the cell bodies. People do patching of axons, but patching, I mean doing recordings from axons, but that's incredibly.
Is that when they get
I bet
a thing and they kind of record when the axon is firing?
Yeah, so they record when the axon is firing. There's different ways to do it. You can sort of attach onto the outside of the cell. You can break open the cell membrane and get inside it and kind of form an electrical continuum with your recording electrode and with the cell. But yeah, that's the giant squid axon, and it's pretty incredible.
It's very cool.
But I think it's just as an adaptation. So for context, we have different thicknesses and sizes of axons in the human body, but typically, and that's particularly the case when it comes to different types of somatosensation, whether it's pain or you have different types of fibers for that, so they conduct at different velocities, different sizes of fibers. But typically in the brain, where we want faster conduction, we have myelination. So these are little sheaths that insulate the neuron and allow the conduction to go faster. But squids aren't able to produce myelin, so their answer to this was just to make an axon that was 1,000 times as big as you do.
That's so funny.
Yeah, it's pretty
Wow,
cool.
that's super
Such
interesting.
little freaks. We're big freaks in this one.
I mean, wow, so squid are truly amazing creatures.
That's what I'm saying. That's why I love them so much. It's just everything you think you know about the physiology or evolution or ecology of something. A cephalopod will come and just blow all those theories out.
Sure.
No pun intended, out of the water.
Are we sure they're not aliens?
I think like altogether the thing that's coming through mostly is that squids are both pretty amazing and pretty damn freaky.
Oh
Yeah.
yeah.
Like
Pretty otherworldly.
cephalopods in general, and I think for these reasons maybe they're not aliens in the context that we think of when we think of aliens, but in many senses, at least relative to us, they are.
Yeah.
They're pretty freaky beings.
They're definitely worthy of many funding and studies. If you're listening, please fund me.
But only with DNA barcoding.
Yes.
That's a pretty
That's a
neat
wrap.
summary
Well,
of squids.
I would like to thank Laura for coming on our podcast.
Absolutely,
that
it's
was
been a
great.
pleasure.
Thank you so much. It was so much fun just to chat about cephalopods.
And I would also like to thank you, the listener, for listening and putting up with our silly nonsense.
Things might get smoother. We're not guaranteeing anything. But yeah, we are endeavouring to get guests on as many of these episodes as possible and get different experts from different fields and different walks of life to discuss their various passions with us.
Yeah, so if you're interested in being on the podcast, please reach out to us. And yeah, thank you.
We'll see you next time.
Till next time. Right, bye.